Hello, I'm Katey Rayner, and I'm here with Minna Kaikkonen, and we just attended the open plenary session where Dr. Kaikkonen gave an excellent presentation. Our session this morning really focused on sort of the genetics of cardiovascular disease and how we can understand the development of disease by the DNA and the genes that we inherit, or not sometimes. Sometimes it's the genes and mutations that we acquire over time as well, which is something I think that we don't often think about when we think about our family risk or our family history of cardiovascular disease. So Minna, so one of the questions that I had for you today was we often hear the words precision medicine, right? And people want to know, what's my genetic risk? What's my score? They want to calculate something. But the work you presented really told us a lot about how the cell context is really important, the disease context is really important, and we can't just look at the DNA and learn everything that we want to learn about how a disease might develop. So do you think we're closer to precision medicine, or are we further away?
Yeah, that's a good question. At the same time, a difficult question. But I would say that we are, of course, closer, because if you can actually now, dissect how each of these risk loci work, you're able to understand better the risk. So let's say we would have 10 people here, and we could categorize that this person, for example, has a risk that acts more through hepatocytes, so liver metabolism, cholesterol metabolism, then that person probably profits more from statin treatment. Then we have another person who has a risk that acts more through the phenotypic transition of small cells. And they would probably profit more from a different sort of treatment.
Right.
So I think that we definitely can get closer in understanding. When we understand disease mechanisms, we can really do better precision medicine. But of course, how to then treat them is the question, because we don't necessarily have drugs that would target, for example, small cell related mechanisms.
And the other presentation that we had today also discussed organoids and how we can actually grow tissues that look a lot more like the organ that has the disease, whether it be liver, whether it be heart, etc. And the vasculature and the blood vessels, which I thought was really, really cool. So then do you see organoids as being something that we, in your work, that you can layer on top of it, which, adds complexity, of course, but then takes along the genetic risk that you were just describing?
Yeah, so definitely they're better than in vitro culture cells, that's for sure. Still, evidently, there is still problems related to kind of the reproducibility of IBS-derived organoids or cells. But I think the unique aspect or advantage of those models is the fact that that you could take a IBS-derived cell from a certain patient. You could derive that into a macrophage, into endothelial cells, into small cells, into hepatocyte. So you would actually be able to see how a certain genetic loci actually acts differently through different cell types, and that's where really the unique aspect is. And I think you can't really do that with much any other models.
Right. Yeah. So. And I think that's kind of the what we learned in the session today was that we can think about genes and cells, which we all do, and we're doing a lot of that at this meeting. But actually, the complexity is so much more than any one of us can appreciate. Yeah. But then if we work together and we look at these things all sort of at the as much as we can at the same time, I think we're going to learn a lot more about disease and eventually treatments, obviously. So Absolutely. It's been a really exciting opening morning for Vascular Discovery.